Hey guys, welcome back to Computational Biology. In this course, we're going to be, this video, we're going to be hopefully wrapping up, we'll see the time limit, wrapping up our input-output for seq data. We'll start by writing some seq or some FASTA files from our seq tools. So let's go ahead and get right on it. Okay, so if you haven't been on here recently, if you don't have it preloaded, import seq, and then we are going to from bio.seq record import seq record. Okay, so rec1 is equal to seq record seq. Why isn't that indenting? Maybe it should be. Well, see if it matters. Let's copy this. So you can put any peptide. If you're going to go on NCBI and grab a peptide, you can put that in there too. It doesn't really matter for our case. 141508338 GB AAK 54648.1. Oh, what happened there? AK, what is it? AK 54648.1. AF 37. You don't just put in gobbledygook here. It doesn't really matter in terms of what we're going to do, but I'm just going to go with what my notes say here. Chalcone synthase from Cucumis sativus. Okay, so let's see if this actually runs. Syntax error. So this is supposed to be indented, I think. So is this. So is this. Oh, there's supposed to be a comma here. That's the problem. There we go. Okay. Rec2 equals seq record seq, and then we're going to put in this code. Oops. That. I say ID equals GI 139196113 GB AK 33142.1. Like I said, just put in whatever you want because we're just going to this is a made up, well, it doesn't matter what you put in for this. Fragaria vesca subspecies rectiata. Oops. Rectiata. Okay. We have too many. Where did I? Comma. Oh, it's because I copied and pasted. So let's get rid of that. Seq is not defined. Capitalize your seq. There we go. And then rec3 equals seq record seq Let's paste in this peptide sequence. My sheet's got, oops. So I'm just gonna paste this whole thing in. And if you are trying to copy me exactly, you can pause it or take a screenshot and do that, or not. There we go, because otherwise you're just gonna watch me type a bunch of letters and, okay. So we have 3 peptide sequences from 3 different species. So we can say my records equals rec 1, rec 2, rec 3. So now we do seek io.write my records, and we're going to call it my example FAA, and we're going to say it's a FASTA file. Seek IO not found. Did we not import? So let's just put right above this, from bio import seek IO. Now let's run it. Okay. So what's going on here? Why? There it is. Okay, it took a minute. I was like, where'd it go? So here we go. So we've now essentially are making kind of like this FASTA file, right, where we had these different sequences all together. This is peptides, right? So it's not amino acids, FAA. So very similar, right? So that's how you write. So if you were doing something like we talked about earlier, where you're like, we're adding random mutations or doing something, or you've manipulated the sequence, you've changed where the start site is, blah, 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 and you want to save your data to like a file, right? That's how we can write it with this. Let's try records equals seek io.parse ls orchid.gbk.gbk. GenBank. Now we're going to say, let's run it. All right, and then we're going to say count equals seek io.write records my example FASTA and then, oops, in the same line, or same box here, we're going to print converted I records to, or records count. Okay, so here we go, as we wrote them as FASTA files. So my example FASTA, so you can see they're written as FASTA as opposed, so they've converted them from peptides to a FASTA file. And then the second line here just is kind of something that you would put if you were kind of like a fail check to see if it converted. So it said it converted all 94 records. Okay. No, actually, I'm incorrect here. So this didn't convert these. Sorry. So because we took records and we said it was this GeneBank ID. So what it did is it took these and saved them as just FASTA files. Right. Right. Okay. Okay. So now let's see, what else do I want to go over? let's do some reverse complements quick here. So from bio, or no, we already did that. Seek IO for record in seek IO dot parse ls orchid dot gbk genbank. All right, so we're writing a loop here. Print record ID. Print record seek reverse complement. Recode record. There we go. Okay, so we're gonna have 94 of these things. So this just prints 94 reverse complements of all of our things. So you see, you can have it, and the cool thing is it like preserves. It knows not to reverse complement like the name of the of each sample, which is convenient. So let's see. Now let's do records equals rec reverse complement ID equals RC and rec ID description equals reverse complement. And we're going to do this for rec and seek io.parse ls orchid.fasta. So what are we doing here? Okay, records. All right, so what are we doing here? What's the point of this code? So what we're doing is, as you can see, so many in here. If you want to not include this in your code, because you're gonna put it in your portfolio, you can delete it. But as you can see here, we just have the gene names, but this is misleading because if we were to print the non-reverse complements, they wouldn't match, right? And unless you like guessed that it was a reverse complement, then you wouldn't know. And so if I had gave you this file before we did this reverse complement thing, and you try to match it up to your LS Orchid gene bank amino acid sequence, or sorry, nucleotide sequence, they wouldn't match. You'd be like, this isn't the same as my data. So what we're going to do is we're going to annotate, right? So what this code does is it says, okay, take the reverse complement, add RC before the name of each of these IDs, so standing for reverse complement, obviously. Add a description that says reverse complement, and then do that for every ID in this file. So now if we print record, I need to put like a video camera on my fingers to see if I'm hitting the R button and it doesn't work consistently, or if I'm just not hitting that button. I'm probably just not hitting because the first time it always works. Anyways, whoa, what's going on here? Why did that happen? Records equals open bracket rec rec reverse complement ID equals RC and rec ID description equals reverse complement for rec and CKO parse ls. Okay, actually let's just look at, all right, blend records. Okay, there's 94 records. Okay, maybe this is supposed to happen. So we'll see, let's see. Let's keep moving on. Records equals rec reverse complement. Wait, okay, yeah. ID equals RC and rec ID description equals reverse complement. We're going to do for rec in seek io dot parse ls orchid dot, did I say orchid earlier? I think I did. FASTA if length of rec is less than 700. So what we're doing here is we're saying if you have a nucleotide sequence that's less than 700, then do this. You can set conditions, right? Parse. Oops. Okay. Description. All right. I'll let you know. It is almost 1 o'clock in the morning that I'm recording this. So give me a little. I'm a night owl, but I'm getting tired. So my typing's already bad, but it's getting worse. Okay, so now if we look at the length of the records, length of the records, not rec. You see we only have 18 because only 18 of them are over 700. So we can selectively, based on like nucleotide length, you can do it based on the genus or whatever, edit your, add descriptions, I guess. Annotate, that's the word, your records. Let's see. All right, let's put this all together as like a complete example. So records equals rec reverse complement ID equals RC. I'll put an underscore there, and rec ID description equals reverse complement for rec and seek io.parse of ls orchid dot FASTA if len rec, oh geez, len rec is, we'll do less than 700. Did I do less than or greater than before? I did less than, okay, good. If it's less than 700, seek io.write, so we're going to write this file, records, rev comp dot FASTA, if, not of, okay. Bioseek IO is no praise. Oh my gosh. Wait. Parse missing one. Oh, wait. Okay, so say I have FASTA on it too. Okay. Jeez Louise. Alright, this is my last video I'm recording tonight. There's like 6 errors in that one. Okay, so we wrote it. We had 18 files, and we should also have a rev comp now that should have 18 FASTA sequences in it. So that's great. So it's kind of compiling everything together. What are we at? Oh, I got some error Windows update. Oh, 19 minutes. Okay, we're gonna quit there. I am gonna record one more video tonight because it's gonna be a really short one, but I want to keep these at 20 minutes and see if my summary came out. It works. I had to like install a bunch of updates. it's been a night. Thanks for watching. I hope you enjoyed this video. We're just about done, and after this next short video that we're going to do, we're going to get into some doing like sequence alignments and things, so like more biology based stuff. The first course we kind of did intro to Python, and this is intro to Python by adding bioinformatics, and then or computational biology, and now we're going to actually get into more computational biology and less about the Python-y managing and things like that. So quick update. I said we're gonna have a short table scrap of our next video. I just started recording that video and the code's all messed up, so we're not gonna have another one. It wasn't important. It was like 2 functions. It was real short, and they were just kind of like, should I put them in, should I not take them in? So this is the last video. So thanks for watching. Please hit like, please hit subscribe, and we're going to get into some sequence alignment in the next section. So we're going to get more and more into the actual biology as opposed to the kind of tools that you need for bioinformatics. So again, thanks for watching, and I'll catch you in the next video. Well, that's awkward.